Hello everyone, myself Hers. I am from Deshbandhu College and today I am here to explain about EMSA, that is the electrophoretic mobility shift assay. Starting with the history of this technique, it was first started in 1981 and the modern shift format, it is commonly called EMSA, was independently described in 1981 by the Gardner and the Revzin and by the Fried and Crothers, who showed how non-denaturing phase can separate the free nucleic acid from the nucleic acid protein complexes. And it has a lot of variants and its extensions. Like starting to its invention, EMSA has been adapted for RNA protein, RNA level free RNA detection, fluorescent readouts, and the different variants are available, which enhances its use. And why it is used? Because it is a method basically combination of simplicity, sensitivity, and direct visual evidence of complex formation, which make it more adopted across the molecular biology, biochemistry, and the regulated element research. So let's talk about EMSA. So what is EMSA? EMSA is a gel-based method which is used to detect and characterize nucleic acid protein interaction or some RNA protein interactions. So it is based on a principle, that is a labeled nucleic acid probe, RNA or the DNA, is incubated with a protein or another RNA. It bonds and form a complex, and the complexes migrate slowly through a non-denaturing polyacrylamide or the agarogel compared to the free probes. So a visible shifted band is produced because of this migration pattern. Now, what are the different elements which are used in these experiments? Are the probes, like the radioactive probes, fluorescent probes, the biotics that are used to give the readouts, and some binding buffers are used, like the salt, pH, comparative tRNA, BSA, glycerol. And what's its role? It basically helps in stabilizes the interactions. And a non-denaturing gel or the short electrophoresis, that it helps in preserving the complexes during the separations. And there are some controls which are used to make it free from any contaminations, like the free probes, specific competitors, non-specific competitors, superset with antibody, and the mutant probes. So what EMSA helps us find? So it helps us in finding the presence or the absence of a stable complex. So it is a qualitative method, and also helps in the specificity of the binding, like the competition or superset assay helps in the finding these things. Now stoichiometric or the multiple complexes, that are different shifted bands, can indicate the distinct complexes, like different complexes are formed if we see the more than one bands. So with careful design, EMSA can be used to estimate the relative affinity, that is the KD, by titrating the protein or the probe. So there are some common variants, like the superset, which add antibody to identify protein in the complex, that is the bigger set. Now the level free EMSA is the staining detection methods that avoids the radioactive probes. So it is useful for the KD measurements. KD is the relative affinity. Fluorescent EMSA are dual level, that is it helps in visualizing the nucleic acid and the protein, and simultaneously using the fluorophores. Now RNA and miRNA and the mRNA EMSA, that is it adapted to show RNA interaction, which is used in the mRNA target validation. Every technique has advantage and its limitations. EMSA also has some advantage and its limitation. Advantages like the it is simple, inexpensive, visual, and it is very high sensitivity. It so result very fast, in that is basically few hours, and it is also flexible, like it works with the crude extracts, purified proteins, or the short RNA or the DNAs. And it has some limitations also, like it is primarily in vitro, that is binding in a gel does not guarantee the regulation in the living cells, and some interactions dissociate during the electrophoresis, that is the weak or the transient complexes which are formed that can be missed. And the labels like the fluorophore, the biotin can sometime after binding, the controls must be essential, and there is no straightforwardly high throughput without adaptation. Start with the research papers which I have referred for this technique. So starting with paper 1, which is basically the rapid preparation of high purity nuclear proteins from a small number of culture cells for use in electrophoretic mobility shift assay. So what are the main goal of the paper? The main goal of the paper was to develop a simple, fast, cost-effective method which is used to prepare a highly purified nuclear protein extracts from a small number of culture cells, so those extracts can be used in the downstream assays. So why EMSA is relevant in this paper? So author optimized the nuclear protein preparation specifically for the EMSA, because EMSA requires the clean nuclear extracts, that is the transcription factors are nuclear, and the small starting material makes conventional large scale preparations impractical. So how EMSA was used? So after preparing the nuclear extracts from the small number of cells, the author performed the EMSA to test the DNA protein binding affinity, and they used an NF-kappa B probe to detect the NF-kappa B DNA binding activity and compared signals from these small scale extracts with signals from standard large scale extracts. So EMSA readouts, that is shift bands corresponding to DNA protein complexes, were used as a functional proof that the small scale extraction preserved native transcription factor activity. How EMSA helped them in the paper? That is the number of the functional validation. So EMSA provided a direct functional readout, that the binding or the no binding is there, that is so the small scale extracts retained active transcription factor, for example NF-kappa B, and also show sensitivity with the low cell numbers. And the EMSA have showed that even with the few cells, the preparation yielded enough active nuclear proteins to take the transcription factor binding, key proof that the method works for the low input samples. And the practical advantage is also there because EMSA are relatively quick and visual. It let the author test multiple samples concurrently to demonstrate the reproducibility and the throughput of the extraction method. Now, the second paper, that is the which is on the topic is validation of miRNA-mRNA interaction by the electrophoretic mobility shift assay. So what is the main goal of the paper? It explores and validates the EMSA as a direct method to test whether a specific microRNA, the miRNA, physically binds its predicted target mRNA sequence at the 3' UTR fragments. So the paper represents the experimental workflow and tests the controls to show specificity. So EMSA basically classically used for DNA protein or the protein RNA complexes, but this paper extends application to miRNA or the mRNA, that is the RNA-RNA interactions, and documents how to run and interpret such assay. So how EMSA was used in this paper? The author designed the short RNA molecules, the mature miRNA, a segment of the target mRNA, 3' UTR, which controls the predicted binding sites, and the various control at the antisense oligo mismatched version of the UTR sequence, and the tables are provided in the paper, which is I will show the reference for that. They incubated miRNA and the target UTR fragments together under binding conditions, then ran non-denaturing polyacrylamide gels to look for mobility shift, that the miRNA bound to UTR runs slower, that is the shifted bands are produced. So the specificity controls included competition with excess unlabeled oligo, use of mismatched UTR sequences, and antisense or the anti-MIR oligonucleotides to disrupt the binding. Each control changes or abolishes the shifted bands if the interaction was specific. What EMSA enabled or demonstrated, how it helped the paper. Number one, it helps in direct physical evidence of the binding, because EMSA produces visible shifted bands corresponding to the miRNA or the UTR complexes, giving direct biochemical evidences that the miRNA can bind predicted target sequence in vitro, and it complements the indirect assays, for example reporter assays. A specificity testing. So author used competition and the mismatch controls in the EMSA, which show the binding was sequence specific. Loss or reduction of shift with mismatched UTR or antisense oligo, it strengthened the claim of a bona fide interaction. Now talk about the overall conclusion of both the papers. So both studies highlights the enduring versatility and the importance of the EMSA technique. That is a powerful biochemical tool for detecting molecular interactions. Liu et al. Demonstrated how EMSA can effectively validate the functionality of the transcriptional factors using the high purity nuclear protein obtained from the limited cell samples, while Sol et al. Explained its applications to RNA interactions, confirming miRNA or the mRNA binding with a strong specificity. Together, this work emphasizes that the EMSA is not only simple, sensitive, and cost effective, but also adaptable to diverse molecular systems, making it an essential technique for studying gene regulation, nucleic acid protein interactions, and post-transcriptional control mechanisms in modern molecular biology. What are the applications of EMSA? It helps in detecting the DNA protein binding, that is the transcriptional factor binding. And how it helps? It provides a simple, direct visual readouts, as the shifted bands are produced that a protein binds to a specific DNA sequence. It maps a sequence specificity, tests mutants, and how it helps compare the binding of the wild type versus the mutated probes, or use the competition with specific versus non-specific oligos to demonstrate sequence specificity. It helps in identify the DNA binding proteins in crude extracts. And how it helps? It using the extracts instead of purified proteins shows whether a cellular extract contains an activity that binds the probe, as a superset antibody can help in identify the factor. Compared binding under the different conditions, that is the cofactor, post-translational state, that is the run parallel binding reactions with or without cofactors, salt, phosphorylation mimics, etc. to see the condition dependent shift. It helps in estimating the relative affinity, that is the semi-quantitative KD. Titrate proteins of the probe and measure the fraction bound at equilibrium to derive binding curves and estimate the KD, that is the relative or the approximate. Last, these are the references which I have followed for making this whole presentations. So that's all. Thank you so much for listening this. Hope it will be helpful.